Neurospora is a genus of Ascomycete fungi. The genus name, meaning nerve spore, refers to the characteristic striations on the spores that resemble axons. The best-known species in this genus is Neurospora crassa, a common model organism in biology. Neurospora intermedia var. Oncomensis is believed to be the only mold belonging to Neurospora which is used in food production, to make oncom. Neurospora species are molds with broadly spreading colonies, with abundant production of ascomata. Ascomata are superficial or immersed, parathecial and osteolate or cleistothecial and non-osteolate, hairy or glabrous, dark-colored. Peridium membranaceous, acicylindrical, clavate or subspherical, with a persistent or evanescent wall, usually with a thickened and non-amyloid annular structure at the apex, usually 8-spored. Ascospores broadly fusiform, ellipsoidal, or nearly spherical, unicellular, hyaline to yellowish-brown or olive-brown, becoming dark and opaque at maturity. Ascospore wall with longitudinal ribs or pitted, occasionally nearly smooth, 1 to 2, but rarely up to 12. Germ pores disposed at the ends of the ascospores, gelatinous sheaths, or appendages are absent. Anamorphs are known in only a relatively small number of species, which belong to the fungi imperfecti genus Chrysonellia. The type species of the genus is Neurospora cytophila shear. The former genera Gelus and Neurospora and Neurospora are closely related and not resolved as monophyletic groups, thus the former genus is nowadays included in Neurospora. Neurospora is widely used in genetics as a model organism, especially in crassa, because it is quickly reproducing, is easy to culture, and can survive on minimal media, inorganic salts, glucose, water, and biotin in agar. The first studies of sexual reproduction in Neurospora were made by B. O. Dodge. Neurospora was later used by George Wells Beadle and Edward Laurie Tatum in X-ray mutation experiments in order to discover mutants that would differ in nutritional requirements. The results of their experiments led them to the one gene one enzyme hypothesis, in which they postulated that every enzyme was encoded with its own gene. Research with Neurospora is reported semi-annually at the Neurospora meeting at Asilomar, California, coordinated by the Fungal Genetics Stock Center. Mutant and wild-type strains of Neurospora are available from the FGSC. The FGSC also publishes the Fungal Genetics Reports. Important people in Neurospora research: Bernard Ogilvy Dodge, 1872–1960. George Beadle, Nobel Prize in Physiology or Medicine, 1958. Edward Tatum, Nobel Prize in Physiology or Medicine, 1958. Hester Lederberg, Norman Giles, David Perkins, Robert Metzenberg, Norman Horowitz, Herschel K. Mitchell, Mary B. Mitchell, Martha Marrow. In the heterophilic species Neurospora crassa, interaction of haploid strains of opposite mating type is necessary for the occurrence of sexual reproduction and the production of ascospores by meiosis. Ascospores then restore haploid individuals of either mating type. The life cycle phase is thus predominantly haploid. However, upon mating, the nuclei do not immediately fuse. Karyogamy is delayed until the very onset of meiosis. The resulting mycelium is called a heterokaryon, and is neither diploid nor haploid. The genus Neurospora also includes homophilic species in which a single haploid individual carries both mating type loci and can undergo self-fertilization leading to meiosis and sexual reproduction. Neurospora africana is an example of such a species. Additionally, some Neurospora species are said pseudo-homophilic. They carry both mating types, but in separate nuclei in the same individual. Two haploid nuclei originating from the same meiosis are packaged into one ascospore. The individual is thus permanently heterokaryotic. Examples of this mating system include Neurospora tetrasperma and Neurospora tetraspora. Because heterophilic species necessarily undergo some degree of outcrossing, they may benefit from a higher efficiency of selection because of higher effective recombination rates. In contrast, pseudo-homophilic and homophilic species do not outcross, or rarely, and do not experience these benefits. In homophilics, a reduced efficiency of negative selection has been shown. However, both hetero and pseudo-homophilic species benefit from the masking of deleterious recessive alleles in the heterokaryotic phase. In addition, all species derive the benefits of meiosis that include the removal of stress-induced DNA damages by homologous recombinational repair, and the formation of stress-resistant ascospores.